Hey, Professor Gould again. In this section, we're actually going to start getting into the nitty-gritty of the cell cycle. I'm not going to do mitosis yet. We're not there yet. We're going to talk about these parts of the life cycle: interphase, G1, S phase, and G2. What do those mean? Hang on, we'll get there. Okay. All right. So cells spend most of their life not replicating themselves, like just like every other organism on the well, they don't spend their whole life doing mitosis. They have things to do. Like if you think about, a cell in the liver, it's got to make bile. It's got to check and make sure that red blood cells going by are healthy. It's got to break down toxins. It's got a lot of work to do. It can't just spend all of its time replicating, or then it wouldn't get anything done. So the time that the cells are not doing mitosis, the rest of the cycle is called interphase. And interphase is divided into 3 phases: G1, S, and G2. So we're going to break those down, see what happens during each of these phases, and then finally we'll get to mitosis. Mitosis, that's when the cell is actually going to duplicate itself. All right, so interphase, like I said, divided into 3 phases. The first one is called G1, or the gap 1 phase. And the first scientists who discovered this, they called it the gap phase because it looked like the cell wasn't doing anything because it wasn't doing mitosis and it wasn't copying its DNA. So it turned out it was building up its energy reserve so it would have energy to do the DNA copying that it needs to do. It's got to build up, got to have tons of ATP ready, got to have tons of glucose so it can do more ATP synthesis, got to build up the cytoplasm because the cell needs to be big because it's going to divide itself in 2. That's how it's going to make a copy of itself. And then it's going to synthesize all of the proteins that it needs to replicate the DNA. It needs lots of proteins, and it also needs lots of nucleotides. Remember that DNA is a polymer of nucleotides. So if we're going to make more DNA, we have to have a bunch of nucleotides at the ready to get copied, right? So how do we control, or how does the cell control moving from one phase of the cell cycle into the next? Of course, there's a way to regulate this, because if there's no way to regulate it, then the cell just willy-nilly splits itself in 2. That's not gonna really help anybody. We're gonna talk later about what happens when that happens. All right, each phase, or not each phase, almost all the phases, include what's called a checkpoint, which is essentially a check yourself before you wreck yourself point, where there are actually cell proteins that monitor the cell and figure out whether it's ready to go into the next phase. So the first checkpoint happens at the end of this G1 phase, and it's called, cleverly enough, the G1 checkpoint. Now, this is a restriction checkpoint, meaning that if the cell pauses at this point, then the others won't happen, mitosis won't happen. Once the cell gets past G1, then it will proceed to the others, okay? The other checkpoints can slow things down, the G2 checkpoint, or it can destroy the cell. We'll talk about that. But it can't, the G2 is not going to stop everything. The only way it stops everything is by destroying the cell. Okay, so at the G1 checkpoint, the cell is going to check that it has sufficient energy reserves, enough proteins, and enough nucleotides. It's going to make sure that the cell is big enough, and it will have checked the DNA, literally read the DNA to make sure that there aren't any gaps, there aren't any breaks, that the DNA is all properly packaged and undamaged. All right, next up, S phase. S for synthesis. So at this point, the cell is going to synthesize DNA. We call it DNA replication or DNA duplication, okay? Each chromosome is going to copy all of its DNA. And if you think about it, this makes perfect sense, right? If you're going to make a second cell, the second cell has to be an exact copy of the first cell. Okay, let's imagine that. Let's go back to our cell in the liver. We don't need a different cell in the liver. We need another liver cell. and it has to have a full set of DNA. Well, each cell has one full set of DNA. It doesn't have an extra lying around. So it's actually got to make an extra set and then keep it organized so that during mitosis, each resulting cell, what we call the daughter cells, are going to have a full set of that DNA. All right, so how do we do this duplication? Well, remember, mom's always going to tell you, now duplicate your DNA before dividing. And this is the little baby cell saying, but mom, because of course kids never want to do what they're supposed to do. All right. Surprise, surprise, we have an enzyme called DNA polymerase, which makes a polymer of DNA, which is what DNA is. So it's going to go along, and it's actually going to split the DNA double helix. Remember that in the middle of the double helix, there are bonds between the nucleotides, between the bases, in the middle of the base pair. And remember that on each side, these nucleotides are complementary base paired to each other. So A is always bound to a T, T on one is bound to an A on the other, C, G, G, C. So DNA polymerase opens up the DNA strand and then makes a new complementary base pair. And it does that to both strands. So we've got where we had this one bound to this one, we're going to add a new nucleotide to each one of these so that they both end up as a full double helix. Now, I am going to provide a link to this video, actually an updated version of this DNA replication by the Amoeba sisters, because they just do a really good job of explaining this. So I'm going to kind of jump through how DNA polymerase works. But here's the thing. DNA polymerase only works in one direction, 5' to 3'. So that has to do with which oxygen on the sugar molecule that is part of the nucleotide, 5' to 3'. So on one end, this works great. It can just start at the 5' and at the 3' end, bind to 5', keep going, blah, 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 blah. On the other end, it has to wait until the double helix opens before it can bind and then proceed for a while and then jump back, bind again, and proceed for a while. This means that there are gaps on one strand. So there's a special molecule called ligase that comes through and closes up those gaps, adding in the missing nucleotides. Okay, so we end up with two complete strands of DNA, and each one is half old DNA from the original cell and half new DNA. Okay, all right. Problem. When DNA polymerase gets to the end of the DNA, one side can go all the way to the end, just keeps going until it falls off. The other, remember, it has to start at one point and then work backward, which means that there's a point that it can't get to. And so we end up with these little ends that the DNA polymerase can't get to. And so in order to protect our chromosomes, we have these repetitive ends called telomeres. And these are non-coding strands, so they don't actually code for a protein, right? It's just a tail on the chromosome to protect the coding portions that actually code for a protein. But if this just sits here like this, then ligase comes along and says, oop, I got a gap. I got to fix this. Or others come along and say, oop, I got to fix this, and actually end up cutting off the extra DNA here, thinking it's extra. So these telomeres, they actually fold over themselves so they don't look broken. So this little end here is going to fold complementary base pair with itself, and the DNA is like, oh, look, I am complete. Ligase doesn't try to fix it. Okay? That is one way that chromosomes fix themselves. Not all chromosomes have a way to rebuild the telomere. And that's one of the things that we think is tied to aging, is the shortening of the telomeres on your chromosomes. Some cells have an enzyme called telomerase, which makes telomeres. So it actually rebuilds the end of the telomere, adds these nucleotides so that the telomeres never shorten. That happens. Where would you expect to find this? In cells that do mitosis all the time. So there are some cells, like the cells at the basal cells in your skin, in your epidermis, which is the top layer of your skin. They're constantly they just go through the cell cycle like all the time to replace themselves, to make a new cell, because your old skin cells are constantly sloughing off because they're dead. That's one of the protections you have against the environment. Those cells have telomerase to repair the telomeres so they don't age. I know, how cool is that? All right. Once DNA replication is done, each chromosome started out as one strand. Okay, this is a condensed double helix of DNA. Now that we've made a duplicate, we have the chromosome looking in this metaphase chromosome structure that we're used to seeing, which is two strands of DNA, each of which is a double helix. These are now called sister chromatids. So this is one chromatid here, and this is one chromatid here. Together, because they are bound together by the structure at the middle called a centromere, they are called one chromosome. When they split, they will each become their own chromosome. Until then, they are sister chromatids bound together as a single chromosome. Okay, that can be a little confusing, so make sure that you just repeat that to yourself over and over. The centromere is this whole region that has some proteins to hold the strands together and a little bit of the DNA. Okay, in your textbook, it calls this the Centromeric Region, which just means the region where the centromere is, and the centromere itself is a region. Really wish they hadn't used that term. Okay, so within the centromere, we're going to build up these structures called kinetochores. That's actually going to happen. These kinetochores building up is going to happen during mitosis. Remember, kine means movement. So these are going to be involved in the movement of the sister chromatids away from each other. I know, that's kind of cool, right? Okay, so S phase doesn't itself have a checkpoint. Once the DNA is copied, while it is being copied, there are other molecules that come along and proofread the DNA to make sure that an A is bound to a T. If they find an A bound to a G, they take out whatever the new nucleotide is, put in the correct one. Okay, and so that's going to happen constantly during DNA synthesis. So there's no checkpoint at the end of this. The cell's just going to go straight on into G2 once DNA synthesis is completed. All right, one other thing that happens during S phase is an important thing that happens in the cytoplasm. Okay, so remember, the DNA is getting duplicated within the nucleus. The nucleus is still intact. The other thing is there's an organelle out here that we did not talk about when we talked about the organelles, and that is called a centrosome. Now, there are going to be a bunch of things called centro something or others. Okay, we got centrosomes, we got centrioles, we got centromeres. So, way to keep yourself organized in your brain, remember that some or soma means a body. And so this is a body in the cell, an organelle, that is involved in this whole process with all the centro things. Okay? Now, this is in animal cells. This contains these two microtubule structures called centrioles. And oddly enough, it seems like this is super important in animal cells for mitosis to happen correctly, but these don't exist in plant cells, so okay, whatever, I don't know. All right, next up, we go through the G2 phase, second gap phase or gap 2 phase, and now the cell is again going to build up energy reserves because mitosis is going to take a lot of energy. and it's going to build up all of the proteins for chromosome movement. Okay, so it's going to need some extra proteins around. It's also going to duplicate all of the organelles in the cell. So it's going to duplicate all the Golgi, all the endoplasmic reticulum, make a new set of ribosomes. All of those things are going to be duplicated so that when the cell starts mitosis, it is absolutely ready with double everything. So now all it's going to have to do is organize everything. You go to one end, you go to the other, and make sure that each of the resulting cells gets a full set of organelles and a full set of DNA. So we do have a checkpoint. The checkpoint makes sure that all the chromosomes have been fully replicated, makes sure again that the DNA is undamaged, and checks if there are any errors in the replication of DNA. It can either fix them or it can cause apoptosis. Okay. All right. So that's the G2 checkpoint. One more check yourself before you head into mitosis, because if you don't check yourself before mitosis, you're going to wreck yourself. So next up, we're going to talk about a little more about how all of this is regulated.